Welcome friends to my YouTube channel. Today I'll be talking on very important topic which is called gene cloning and recombinant DNA technology. I will not discuss the whole detailed lecture on the gene cloning, which may take some time, but I'll just try to solve the problem of students, which many times students ask me. Many times when the students attempt to perform gene cloning and for purpose of any insertion of a gene into the vector, they fail to get the results through transformations, and recombinants may not get onto the plate. It happened many times, especially when students attempt first time or second time. So what is the major problem behind that? Where is it, how to troubleshoot it, and where to hit the basically to resolve this issue? This is what I'll be discussing today. So as we all know that when you clone a gene, it is the plasmid and the gene that has to be mixed together, and to insert this gene, or we call the insert, into the vector, so we call the construct. Now when you make a construct into the vector and transform, right, you will get the transformants on the selection media. It may be blue and white colonies, or it may be the plasmid ampicillin based selection or antibiotic based selection. It depends on how did you clone the gene. Now, the very first reason of not getting the recombinant colonies, right, or even sometimes transformants, is that the digestion. So if you are not able to properly digest your insert or digest your gene, in other words, which has been amplified through PCR, then you cannot insert into the vector, and there's no recombinant selection, right? First I will tell whether you will get the recombinant or you will get the transformant. So there's a difference between transformants and recombinants. Sometimes if you're not having the blue and white selection, then you might confuse with the transformants with the recombinant. So transformant means you have somehow plasmids part is digested or they're ligated, but your insert could not digest and could not be inserted. I mean, the gene is not inserted into the vector, and there's a not proper ligation. It is the self-ligation of the plasmid after digestion. Sometimes you may not have digestion of even both the product, plasmid as well as the gene. So it's the very first thing that you have to look into the digestion. So you have to check the enzymes, working or not. So you have to rule out the possibility and ensure that the enzyme is working. Sometimes you may have a single enzyme on both the ends, or sometimes you have a two enzyme, one enzyme at one end, another enzyme in another end. So in both the cases, you have to ensure the proper digestion, and that can be checked through the, after digestion, you run the gel and you can find the linearized fragment in case of vector. But of course the replicon, the amplicon or the gene cannot be find. I mean, you cannot using through gel, you cannot find whether it is digested or not. But while you insert is mixed with the vector, cut vector, that is digested vector, then you may have chance of proper ligation. So ligation is not a problem, or not a much great problem rather, but the digestion is a problem. So always you try to first find out whether your gene and your vector is digested or not. If not, then you have to again optimize the condition for digestion, increase the time, right? Change the enzyme. Sometimes companies may also affect. So once you ensure your digestion is done, then you go for ligation. And ligation is always easy compared than digestion. Digestion may have a problem many times. Ligation could also have a problem, but I think ligation, once you have digested, there's a chance of ligation is always there if it is a sticky end. If it is a blunt end, then again you can have some other options can be taken, but I think blunt end can also be ligated without any problem. Once you ligate it whether at 4-degree temperature or whether at a overnight 16-degree temperature, or there's a different procedure of ligation overnight or a few hours and so on. You have to optimize the ligation condition. Once you ensure the product is ligated, your insert has been inserted into the construct, into the vector, and you have made a construct, then you transform it, right, into component cells. You have to make the component cells. Whatever cells you can take, there's a lot of many component cells are available. You can make commercially available component cells, or you can make the component cell in the lab. Component cell should be fresh, right? It's not like days and months old component cells you can again revive. No, I always prefer the component cell should be freshly made. And once you have component cells, you go for transformation. After transformation, you select on the if it is axial media and it is a beta galactosidase insertion, that is a blue and white colony, then you have to use axial media. Otherwise, simply ampicillin selection can be made. But you may have, if you're not having the blue and white insertion, blue and white selection, then you may have a problem in comparing the white and blue colony. If not there, then you may have a chance of like confusion with the recombinant and transformants. So when you pick up the colony from the plates, ampicillin plate, you may have a recombinant and you may not have a recombinant. So to check primarily to check the recombinant, simply you go for digestion and you can just check the fall off. And through fall off, you can see the bands being cut from same digestion enzyme restriction enzymes into two bands. One is plasmid band, another is the your insert. So that way you can calculate the size, and you can compare the size, and you can say, okay, my insert is there, so this might be the clone. Then finally you have to confirm with the sequencing. So the major problem here is the digestion. So restriction digestion should be very much ensured that you have done. You can change the enzymes, you can optimize the condition for enzymes. Suppose you are following the papers, sometimes papers from different labs may have different conditions. So you have to change the conditions, you have to optimize the conditions, and once you confirm the digestion is there, then go for ligations and then transformations, and you get the clone. So this is a simple troubleshoot video